So,
Uh-huh.
I'm just, so thank you so much for joining. Okay.
Thank
And
you. Thank you.
yeah, I'll formally introduce myself today because last three webinars we didn't get the time. So my name is Dr. Smita. I am also a physician, but I specialize in pharmacology. So I was into clinical research before. In way back in 2013, almost 10 years back, I started my own company. I'm into B2B communications. I work for pharma companies. I work for medical devices company. I prefer B2B related work. I create communication. I convey the story of brand to their end consumer. So that's what I do on day-to-day basis. I have a team in Bangalore and I have a team in Pune, and it's going good. And regarding the longevity, it's my passion. I started researching on it after I started, working on my fitness, and after started looking at like the science of aging. I have a strong family history of hypertension, stroke, diabetes, and everything. And being a doctor, we all know that this is what is important, right? If you have a family history, you need to take care of yourself first. So that's where my journey started. Started researching, then got to know more and more things from Darshit, and then we started all these webinars with Decode Age. I work very closely with the team and learn a lot, as well as quite many exciting things that we all are doing together. So that's my background, and today we'll have almost sort of like a one-to-one session with Darshit. Darshit, could you please introduce yourself?
Uh, Thank you, Dr. Smita. So yes, I'm Darshit, like we all know by far, and I have done my drug design and metagenomics as background in research and education, and I started Decode Age a couple of years back, considering that there was a definite lack in the longevity sciences awareness and even research in India. So there's a lot going about in the world, be it the Harvards and the Cambridges of it, or the big companies and the Mayo clinics and all. So there was definitely a lot of science. There was a lot of progress in the past couple of decades, and it was time that India had a glance of it. And that's when I got together with Parth and Rakesh out here, who are my co-founders, and helped me take up this journey to make 100 years of healthy life a reality. And why 100 years of healthy life? That's the vision, of course. But what's the problem that is coming across is that people are experiencing a much lower health span, which is the time between 40 to 70, which is the time when a person lives with a disease or medicines. That time has to be reduced. If a person stays healthy for a longer period of time, disease free for a longer period of time, they have definite capability of going to a longer extent as well in terms of lifespans. But now, so far, throughout the medical and healthcare, segments in the past 4 or 5 decades, we have been constantly targeting the chronic diseases, and we have definitely gained a lot of insights out of it, and we have managed most of them. But the underlying sign or the underlying reason why these diseases were always developing was aging. And finally, we have some good insights, some good theories, some good hypotheses to prove in the terms of aging. In fact, most of them have now been proved as well. And 9 of those proved hypotheses kind of will be discussing today in this webinar, that is the 9 hallmarks of aging. Basically, a hallmark is anything that indicates that, okay, your body is going through a certain kind of physiology or pathophysiology. In terms of aging, it's these 9 hallmarks. And the interesting thing about all of them is it is at a cellular level, which means the very base root or the grassroots level of where the aging starts. If we see there's an organism where we feel the effect as a person, right? If anything goes wrong with our body, we feel it in our body. That's at an organism level. Within the organism are many organs. Within these organs are many tissues. And within these tissues are the cells. Now, cells is what we really focus on when we come down to hallmarks of aging, because that's where the cascade of reaction starts. And that's what we are going to be discussing today. So I'll give it back to Dr. Smita, and I'll thank her for, helping us out with everything.
Darshit, thank you so much. And I think formal introduction is always required. We try to skip it every time. Another thing is, being a physician, most of the chronic diseases, we try to treat them or we try to intervene only when there is already the damage that happened. But the aging related science that has been evolving, as well as so much of research that is happening, help us to get into the phase where probably we'll be able to prevent many of the chronic diseases. Of course, it's at a very, very initial stage. My personal interest lies in that area. And that's the reason sometimes it's very important to go back to basics, understand what is happening at the cellular level. Of course, your interest, my interest will be always to find out how this is becoming a risk factor for the diseases that we have. But when even I started, so I've done my MBBS, my MD, but even, and there is a huge gap after I left my college. But when I started researching on longevity and hallmarks of aging, honestly, I took like enough time to even get to know what all these nine hallmarks, how they are interlinked, whether they are interlinked or not, where exactly it is starting. I have to go back to go basics. So it's very important that today, the time that we are investing to understand at molecular level, each hallmark we are trying to simplify it. So that's it. What my requirement today is, try to simplify as much as possible, because you have spent more time on these nine hallmarks than me, spent in whatever the research time it is. So we'll start with all the nine hallmarks. So everybody, if you can see this diagram that is there. So this actually, the whole discussion started in 2013 when there was one publication in the journal Cell, where they have said that, okay, these are the nine hallmarks of aging. Now this became the milestone, and after that, quite a much research that happened to find out how these each hallmark is affecting the aging process. Can we modify those and come up with some kind of a supplements or intervention? These are lifestyle interventions as well. So there's so much work happened after this, and this is the basic. So I want to start with the first is genomic instability. Okay. So this word, Darshit, I want you to help me to understand in a very, very simplified way. I want to go back and explain it to my daughter and want her to start the lifestyle in that way. So explain me at that level. Not too much, she's smart. So you're on mute. Are you on mute? Yes, you are.
Yeah. Right. Of course, Dr. Smith. And I understand how important it is to, really understand in a very layman level, because it clarifies things a lot. And today when we talk about genomic instability, right? So we just talked about how organism is built by cells, which is at a very cellular level. Now, if we consider cell like a kingdom, every kingdom has a capital city, and the capital city is the most protected place in the entire kingdom. So that is what is nucleus for us. In this capital city, we will always find all the important stuff that is there about the kingdom, be it the documents, be it their, treasury or anything. Similarly, within the cell, within the nucleus, there is this treasury of information about the entire kingdom, and not just the cell, about the entire organism. This treasury of information is basically genome. And when genome is stable, that means it is able to give out right information. We can go into the genome, we can get the right information, do the right thing in the body, and put it back. That is what a stable formation of genome would be. But now there is instability with aging. And how does it come? There are a lot of factors. Now within the genome, there is this big ball of condensed DNA, which we call the entire genetic material. Together, this makes up the genome. Basically, it would look something like this. This is a condensed form of DNA that is present inside the cell. Now, this formation is very important. As soon as there is some defect into this, things start going wrong in your body. That is what instability is. Now, what kind of problems there might be for this to happen, right? First of all, if you want to read a book, you will have to open it. Similarly, when you want to read the information from a genome, you have to open the genome. Basically, open the DNA. That is what DNA replication is. A DNA which is once wound up, opens up, creates a copy, then comes back, winds itself up again, and goes into the information center. This is what happens to read the information out of the genome. Now, the problems that could happen. If the DNA opens up, what if it cannot bind back? That is the first problem. Secondly, what if this genome is constantly being attacked by different factors? There are two factors now. Endogenous, something that we cannot control. Endogenous is something, like I said, when DNA opens up, what if it cannot wind back up, right? So that is one of the endogenous effects. But what are the exogenous effects? These are the toxins, the mitochondrial oxidative stress, for which we take antioxidants basically. That, the UV rays, when all these come, it slices up DNA. It literally breaks parts of DNA. Now, the good part is we can rebuild it by our natural systems itself. Within that capital city are the right people who can go and repair the information. And they do it for the longest period of time. But even they need something to repair it. When a building breaks, you need something to repair it back. Maybe a cement, maybe a block of brick or something. That block of brick is in scarcity as we age. When we start aging, the bricks and the cement that our body has reduces, and which is what, and eventually the repair systems cannot get the right raw material to repair your DNA. So that is how genomic instability starts as we age. And it has been often correlated to many diseases, like because genomics instability is not something that is new. Although it was compiled as a hallmark of aging in 2013, it is something we know about for the past 40, 50 years now. So yeah, to the crux of it, that is what genomic instability would be.
So my question here is, see, we are talking at the cell level, right? So I have different cells in all over my body, right? Like I have parietal cells in my stomach, I have muscle cells, I have cardiac cells, I have cells, neural cells. So, and this information storage, the library that is there, it's available in each and every cell.
Right.
So the change can happen anywhere. I mean, the damage can happen anywhere in your body.
Exactly.
Perfect. Okay. I think it's clear to me regarding genomic instability, and your shoelace is helpful. You think I should ask the questions during the thing, like if anybody has any doubt regarding that, or we can move?
Absolutely.
Okay. So if anybody is not able to, correct. If anybody is not able to understand or have any specific question regarding genomic instability, not the generic for other thing, they can, I think, drop a question into the chat box, and we'll address that. Okay, we'll not wait for it because we have very less time. So moving to the second, I'm not going as per this diagram, moving to the second hallmark, that is epigenetic alteration, right? So what exactly it is? Is it next stage to the genomic instability, or it is linked with that, or how exactly it is affecting?
Right. So we just, like, of course, the genomic and the epigenetic, the pre and the post of the two words would pretty much tell you that they are surrounding each other. Now, so far in genomic instability, we considered. Now, when I open up this, yes, I think Padma has a question, and I think this question will be better answered when we are discussing the mitochondrial dysfunction. So I hope you stay with us, Padma.
Okay.
Yes.
We'll address this, Padma, when we talk about the fourth. So after two more hallmarks.
Right. Okay. So now, what exactly is epigenetic alteration? Now, there are two types, in fact. One is the entire chromosome, or the entire genetic material that we just saw as a ball, is made of DNA. And DNA is a double helix, basically two shoelaces wound up about each other. That is a double helix. Now, this DNA has a certain pattern. If I have to come and read this DNA for the information that I need, there will be a molecule that comes around, reads the information, and goes back. What if this information is blocked? Let's say there is something on it, right? So that is kind of an epigenetic alteration. Why epigenetic is because it's not related to the genome itself, but something from the outside. Basically, when an environment affects the gene, it becomes an epigenetic problem. Now, this is one. Now, secondly, if I have to keep the shoelace wound up properly into that ball that we just saw, I cannot just crush it like this, because when I crush it like this and open it, it will open it up again. What I rather do is I twist it properly. And when I twist it, I see these rounds coming up out here, right? Every time I twist it very, very tightly, right? Here I'm trying to twist it very tightly. And it will become something like these balls. You see some small wound up DNA. Now this has to be maintained, this entire ball. And for that, we have something called a histone. It is basically something like this. If this is wound up about my finger, then my finger is the histone and the surrounding part is the DNA. That is how it's wound up tightly. If this finger goes off or if this finger is modified, then this part opens up again. That means there is a problem in the genomic instability. This maintenance of histone, this finger and its structure and the way it wounds up the DNA around itself to protect itself from any kind of damage is basically a histone. And when histone is modified, there is an epigenetic change. That epigenetic change can open up the DNA and eventually lead to genomic instability. So basically the major difference between genomic instability and epigenetic is genomic instability deals with the DNA or the genome itself, whereas epigenetics is something that affects it from the outside. Is the major difference why these two hallmarks are segregated. But yes, epigenetic leads to genomic instability.
So while you are discussing, I'm able to, in my own visualization, I'm thinking as if there is a library where there is a code, okay, and the walls are basically the epigenetic, material, and the alteration is for the room, okay, and the code is basically in the books. So that way I'm able to visualize it when you are explaining with the shoelace.
Exactly.
I guess if any question regarding epigenetic alteration, it's super clear. I am able to explain it to my daughter for sure. Please put it into the chat box. We'll go to your favorite, telomere attrition.
Right. Now, this is the most interesting part. Now, when we see the entire DNA like this, right, I have wound it up at the end. These are the four corners, or what we call the telomeres. These are the end parts of your DNA. Now, we see every time a shoelace has an egglet. Why do they put it up? Because if this egglet is not there, the DNA will start, or the lace will start opening up, basically. Similarly, there's a telomere at the end. It's basically a cap to protect your DNA. As soon as this is gone, right, the DNA starts opening up. This knot at the end will open up, and then you see how the shoelace is unfurled. It will be literally two different strands. Now, this telomere is very important. Why? Because long back in around 1950s, there was a scientist called Dr. Leonard Hayflick. He saw that when he was growing the cells, his cells couldn't divide after a certain point of time. What could be the reason for that? For 30 years, he lived almost in a mystery. He couldn't know why that was happening. 30 years later, someone took a Nobel Prize to discover why this was happening. This was happening because of the telomeres. What Dr. Hayflick could confer was that every cell could divide around 40 to 45 times. But what gives your cells the signal to stop dividing after that? Well, it was the telomere. Because every time a cell divides, a part of this is cut off. Second time the cell divides, second part is cut off. Third time, third part is cut off. Eventually, at the end of 40 cell divisions, there'll be no cap. And now the DNA start unfurling. The cell cannot read the information. The kingdom collapses. That is how the cell collapses. Now, telomere attrition is basically one of the most recognized hallmarks of aging, because if telomere is gone, there is again genomic instability. And with genomic instability, everything ruptures. So is why telomere is such an important part and has been included in the hallmarks of aging section.
And this effect that you are talking about, it's given in this book, the one that you recommended me. This is written by the same person who actually received the Nobel Prize, doctor. But usually we think that doctors will write very boring books, but it's exactly opposite. This book is so interesting, so simplified, and so nicely everything is explained. And another great thing about this book is the first half of the book will talk about all the science and basics and everything. And then it will give the protocol, the formula, that what all interventions you can do in your day-to-day life that will help you to, have the longer telomere or the long life. Okay. In other words, it's a wonderful book. Wonderful. I'm recommending everybody who's reading it. And Darshit, thank you so much. And the effect that you are talking about, it's very nicely, the story has been given in the same book, which talks about they started, the research in the lab, and they were running short of the space because the cells were dividing so much. And one point of time, they thought that how we are going to do this experiment. And one point of time, the cells stopped dividing. Okay, and that's where they recognize that. So it's very nicely. Also, it's the same reference has been given in this book as well. So yes, and I'm reading a lot about telomere attrition. It's very interesting, but I think equally interesting is, especially for the researchers, is mitochondrial dysfunction. So what's happening in that area and what exactly happens over there?
So basically, long back, if we take ourselves to 4 billion years back, when the earth has just cooled down, there is water, there is just water, and then there is minerals. Now at this point of time, the first organism that came up was a bacteria, a very rudimentary bacteria, right? This guy could live. It had basic bioorganic molecules which can keep it alive, but it did not have one thing that could still make it call living, and that was energy. While there was this other guy who was developing at the same time, who had energy, but who could not do anything else except producing energy. So what these two guys decided was, let's make a collaboration. Let's become one so that we can start living together. And that's what actually happened. The bacteria that could live took mitochondria within itself. So now it could live and produce energy, which is the definition of a living being. Movement, energy, and the biochemical processes within the organism. These three started happening when these two came together. Now that energy producing bacteria was basically mitochondria. It happened long back and it has still stayed with us. And why? How can we say that these were two different cells? Maybe they evolved together, but no. Like we said, that the entire information for the entire kingdom stays in the capital city. But there is a bit of information that is there in the mitochondria as well. Mitochondria has its own DNA. That's the only starking feature about the cellular kingdom, because no other part in the cell has its own DNA except the mitochondria. But it is also not as much protected as the nucleus, because the nucleus has evolved to protect the information. Now it is prone to attack. Now where does this attack come from? Within itself. Why? Because when we talk about nuclear reactors, that gives the energy for different electricity and all the processes that are happening in the society. These nuclear power plants are most prone to fires and blasts. Similarly, mitochondria is basically the energy producing center in your cell. So it is also most prone to fires and blast. But what kind of blast? What kind of fires? Well, there is a chain reaction that is going inside the mitochondria. Just like in the nuclear power plant, there are a series of processing happen to generate energy. Similarly, in mitochondria, there is a process, which is called Oxford system. In other language, it is also called the respiratory chain. It's a 5-step process. If this process is not done properly with the right levels of safety, it will lead to fires or blast, which we call the oxidative stress, which needs the antioxidants, right? Now, this oxidative stress that is generated and the fires and blasts that are happening within the mitochondria will damage its own information or its own DNA. Now, within a life cycle, a cell produces many mitochondria within itself. Once that gene is affected or once the DNA is destroyed, the next mitochondria that is going to be produced in the cell will be that mutated. Which means it will not have that part of DNA which has already been destroyed. This leads to more and more mitochondria in your cells with destroyed DNA, and they lead to cellular dysfunction. Now, when the most important job of mitochondria is to take the food, convert it into energy. When it becomes dysfunctional, the food is not converted into energy. And we all know that when food is not converted to energy, a lot many things can happen inside the body. And if the cell is not able to produce its own energy, that means it cannot do its basic processes. When basic processes go down, the cell goes down, basically. So that's the whole theory why mitochondrial dysfunction has been included in the hallmarks as well.
Okay. So, I think along with this, we should put the diagram of the, nucleus, the cell diagram. So I remember I think in 4th grade, we used to have, I don't know which grade now they teach, but 4th grade, we used to have this chapter in this thing, animal cell and plant cell, and the difference between them. And those two diagrams used to be there. And when you are explaining it, just showing me the diagram in my mind, we should have that diagram somewhere over here. Tell me one thing, the mitochondrial dysfunction that you have explained just now, the nuclear blast related thing, and the slide that you show us when we discuss that, is it there in your TED Talk, that slide?
Yes, it is. It is.
Yeah,
You can
so
follow my
for
Telegram.
reference, like his TED talk is there on YouTube, and there he explained this mechanism again with a nice diagram and illustration. So anybody who's interested can see that as well. And I think you said that Padma's question you'll be addressing during the mitochondrial
Oh, yes.
dysfunction, that
Yes.
antioxidant to helps with the repair of DNA or not.
Right. So okay. So how does antioxidant will help with the DNA repair? Well, there is no direct correlation, but what happens is when I say that mitochondria produces blast, and I give a comparison with the nuclear power plant. Let's remember the infamous incident of Chernobyl. When the nuclear power plant blasted, what happened? A lot of radiations were going out, right? Similarly, when a mitochondria blasts off, radiations, something we called molecular bullets, are fired everywhere. These molecular bullets are basically the free radicals that we talk about. We keep seeing on the labels of antioxidants that reduces the free radical formation, counters the free radicals. These free radicals are basically molecular bullets. Now, when they are fired from the mitochondria, they don't care what comes in between. It will go through the nucleus, it will injure your DNA there and there itself, and then it will pass through. Well, antioxidants might not be the best way. It's a 70-year-old theory, actually, that antioxidants help in anti-aging or aging or longevity. But these bullets are produced at a very high number. They're very fast. The antioxidants that you take might not always counter them. The better way to go about it would always be to get the mitochondrial function back in hands. How can we do that is rather controlling those 5-step process that was happening in mitochondria.
That's right.
Now, we very well understand what's happening in that 5-step process. And there is a molecule at 1, molecule at 2, molecule at 3, 4, and 5. The most important problem comes at the molecule number 3 or the complex number 3, where the NAD plus exists. NAD plus is something that is made inside your cell. It goes and helps mitochondria with efficient energy production. But as we age, our capability to produce more and more NAD reduces. Mitochondria does not have enough NAD to utilize, and that complex goes wrong. Now, NAD plus, if we go to, sorry if I'm going to the basic chemistry, but that plus is something about electron transfer. And when that electron transfer doesn't happen properly, that is what leads to oxidative stress. And oxidative stress are those molecular bullets which injure everything. Rather than trying to, it's as simple as that. When someone fires a bullet at you, you cannot just stand in front and show the hand that your hand will try to block it. That is what antioxidant is. It is basically showing a hand to a bullet, expecting the bullet to stop. Well, that's not going to happen. It will go through you, right? So rather just go to the basic, go to the mitochondria, make sure that the bullets are not fired at all.
So a little layman question. So when I see, like I watch television. So when we see that green tea has too many antioxidants, and then if you then it is reducing the oxidative stress and all, it's all very vague, right? You cannot like drink a tea and then expect the oxidative stress to go down, right? Like too vague, I believe.
Well, there is some kind of an effect, but eventually, if we see on a larger term, drinking green tea every day versus the amount of damage that the aging creates is kind of incomparable, right? You might have to drink probably 40, 80 teas a day to even kind of control what's happening inside your body if you're completely depending on that. And it is always damage control is one thing, but if you're not able to control the source of damage also, then it becomes a problem, because body is a complex thing. You cannot keep going damage control every time. You stop the war. You don't try to, let the war happen and try to rebuild the city after that kind of.
Correct. And just a disclaimer, that does not mean that people should go and drink 40 glasses of cups of green tea. There are case studies that publish that there is one person who was on exclusively on the tea diet and gone into toxicity. Okay, so it's a published case study. They're available on PubMed, so that's also not recommended. So everything should be in balance. But yeah, what you mean to say is it's like it's the same thing, like as showing your hand and expecting the bullet to stop. So that's not how it happens. Perfect. So let's move to the next very quickly is loss of proteostasis. So what exactly it is and how it's aging us faster, or is it
So,
affecting age?
right. Now, again, we'll have to go back to industry now, or maybe a factory, right? A factory where goods are produced. Okay. So when you say our cell is basically a factory to produce a lot of proteins every day.
Hmm.
Proteins are the building block of our life, and we need proteins for everything. So cell has a way of making it, right? But the demand of protein is also very high. And a factory
Everywhere.
that functions at a very high pace or at a very high demand does generate a lot of defective goods in a day or something. That is why they have supervisors. What supervisors would do is keep a quality check control mechanism, that okay, if the goods are proper, then it goes into packaging, otherwise they are discarded or segregated away. Good thing about the evolution is that it gave ourselves these different kinds of quality check mechanisms. To be specific, there are three. I won't take the jargon behind it because then it becomes very difficult to follow. So there are 3 or 4 different kinds of these systems. What they do is it's kind of a shape check kind of a thing. A protein is very well folded, right? If we consider any protein, it has to be folded many times, just like we saw with this DNA, right? A protein cannot be like this. It has to be folded in a particular way. Now, after the folds happen, it takes up a certain structure, right? Now, the quality check mechanism, what they will do is they'll pass this structure through that particular formation and see if it is going through properly or not. Just like if you go to Kashmir, right, there'll be a lot of vendors who will come to you with a ring and say that my entire shawl can pass through this ring. Well, if it passes, then it is a Kashmiri wool. If it does not passes, then it is not. So that is what this quality check mechanisms are. It will pass it through the ring. If it passes, well and good. But if it does not, then it will try to destroy that protein there and there itself. Now, with age, that ring is lost. That quality check mechanism is lost, right? And it might not function at the level at which it was doing when you were young. So when that happens, all the misfolded proteins, all the bad structure proteins basically start accumulating in your brain. Not brain, and sorry, inside your cell and inside your body. This protein, which is basically the junk of the body, when it accumulates at a very high extent, it starts taking up the spaces in the cell. It starts taking up the spaces in your tissues and start aggregating there, creating a junk. Now, which cell would like to live in a junkyard? Right? Who would like to live in a junkyard? So things that are around it also starts getting affected. That is what basically loss of proteases is. It's not the ability to lose the making of protein, but rather quality checking the protein to keep only the good ones with you. That is basically what loss of proteases is.
Okay. But it's so wonderful, right? We started with the nucleus. We talked about the genome in the nucleus. We talked about the mitochondria. Now we are talking about the quality mechanism that is there in each cell. And if it is and these are the multiple factors, right? Like beautiful it is. Like it's very interesting.
And
okay,
we should not forget that all this is happening at a cellular level where a pinch of those cells may be in millions of numbers, like that. So it's happening within that cell. Everything is within that cell, and that is where everything is controlled from. So that is what it is.
I think yesterday we were discussing that if half of the world is very much interested in going to Mars, and we see the news over there, there is our kind of a population who wants to go back to the cell level and get fascinated with what all is happening and how it is happening. It's super interesting. Okay, so I hope people have questions regarding this, but to me it is clear, and that's why I moved to the next. Your hallmark is deregulated nutrient sensing. Okay, what exactly it is, and from nucleus to the quality check mechanism, where are we moving now?
Right. So if I take it in the most basic terms, right, a person feels hungry, a person feels thirsty, a person feels something that motivates them to drink water or have food. There are basic sensors in your body that go at an organism level that reminds you of eating, sleeping, or drinking water. Now, similar sensors exist as a very molecular level as well, right? Now, for example, your insulin pathway or the glucose level pathway. When the body has enough glucose, right, there are cellular signals which say that, okay, there is an ample of food, we can start processing. This is what basically the sensing systems are. Similarly, if there is a low energy, then your cell senses that, okay, we need the energy because I can sense that there is low energy. So these sensor mechanisms that, senses nutrient deficiency, that senses energy deficiencies and all that, these start getting affected. They don't function at the optimum level as they used to, back in the day or back in the younger days of a person. This is what basically leads to deregulated nutrient sensing. When some of the pathways involved here are mTOR, right? mTOR is overly expressed, or it is showing overactivity during the aging period of your lives. So what happens is we need to restrict it. Secondly, on the opposite side, there are certain energy sensors which function at a lower level, which says that, sorry, it functions at a higher level. So your body always has a sense that, okay, there is a lot of energy in me, I don't need to produce. But that is not the right thing. When you are aging, the energy levels are dropping down, although those sensors are telling your cells that the energy is high. So that's the basic miscommunication that's happening between the two systems. So when the body is actually experiencing low energy, we have to activate those nutrient sensors, and we need to tell the body that, okay, you need food and you need to start expressing certain kinds of genes. Such kind of misbalance or miscommunication between two systems, right? When you have low energy, but still your body senses its high energy, that is the kind of deregulated nutrient sensing. The sensing word and the deregulated word are kind of related that way. Nutrient because energy is involved.
So these are the nutrients. We are not talking about the nutrition related things.
Yes, nutrients
more
because,
of a molecular level.
Right, at a molecular level, not the nutrition itself. Obviously, nutrition is where the nutrients come from, but nutrients is what affects these pathways.
Correct. And then as we know what exactly is happening, I believe there are quite a many supplements that are available targeting exactly this. So we know that, see, I'm a pharmacologist, so we have medications for everything, right? And we keep coming up with better and better medications if a particular medication have side effects, or if it's not efficacious, or if the dose is not sufficient. So we come up with the better and better formulations. If the a drug is not able to cross the blood-brain barrier and reach, then we come up with the different formulations like that. So there is so much of discovery that happens that helps us to get targeted medications in the market, right? And that's how the whole medicine world works. Similarly, and it's a very regulated one, right? So we go through the complete clinical trial and process. So I believe that if we have reached to around 6th hallmark and we are left with like to discuss 3 more, I believe that the science has evolved enough to have already established trials going on, or established research which talks about the supplements that are either directly affecting or reversing or stopping or agonistic, antagonistic actions for these mechanisms, right? So if you can take 1 minute, not more than that, quickly to tell what all molecules that I have proven studies, okay, that we have with the studies, with data, with science that is backing up about the hallmarks of aging and its effects. What all things are there?
So, of course, if these are the hallmarks of aging, then manipulating or controlling them in some way or the other is going to have some kind of an effect on the aging process. The major goal of any study would be to, reduce the pace at which we are aging. And that's why the science often uses words like slowing down the aging or reversing the aging rather than anti-aging, because you cannot stop the process. Because the process cannot stop. Either it can be slowed down or reversed. So dealing with these hallmarks can actually give you good results on how the aging is accelerated or decelerated in your body. Now, for example, like genomic instability, there we were talking about this guy who repairs the DNA but does not have the enough raw material. So kind of nucleotides, your DNA is made of nucleotides, and that's the raw material it needs. So when something like NMN, which is a precursor to NAD+, right, and NAD+ is what? Dinucleotide. The D in NAD+ stands for dinucleotide. So it's actually a nucleotide donor to that repair system, which eventually rebuilds the DNA. So these kind of theories are developed, hypotheses are developed. There have been clinical trials to show that, okay, there is positive effect. But to really understand it at a cellular level, if it is directly influencing genomic stability, we cannot say that because the science has not developed as of yet. But the outcomes of controlling these are predicted and they have been proved. For example, if you see the same NAD plus, if it is restored in the mitochondrial, then the energy levels of people have increased. Although we cannot say that NAD was there and the NAD that we took was there at the third level or the third process of the compound, but we can still say that, okay, the energy levels have improved. So there is a basic correlation, and sometimes there is a causation about why supplementing with something or the other, or doing certain kind of physical exercises, or doing some meditation, breath work, or anything like any intervention that has been going around is kind of helping us control with the controlled aging. Why does a person following such a strict protocol definitely look younger than the person who is not doing all this at the same age? The simple reason is that these hallmarks are affected at a cellular level in certain way. So there's NMN for genomic instability. We can always see telomere. Much more research is needed that side, but of course, like mitochondria is where most of the research has happened. We see that NAD plus helps there, ubiquinol CoQ10 helps there, right? Similarly, like deregulated nutrient sensing. So there are certain definite pathways like IGFs and the mTORs and the AMPKs. Now we know certain compounds that modulate these pathways. So we can be definitely sure that, okay, using this particular supplement or a compound might directly interplay with the mechanisms that are involved in deregulated nutrient sensing. So I'm sure, yeah, there is a lot of research. There are a lot of clinical trials, in fact, for the outcomes and research on a cellular and animal mouse models to understand the mechanism of how it is happening.
So the research is happening at both the level. And if you see that the diagram that has been displayed over here, we see quite a many natural compounds, and you talk about natural compounds a lot, right? So we have curcumin, aron, haldi, we have ginger extract, and these are also clinical trial based proven that they have effects on aging and slow down the effects of aging. So tell me a little more about them.
So of course, like curcumin is an age-old thing we have kept hearing about forever now, and the clinical trials have definitely shown outcomes that, okay, whatever research has been done has been translated to humans. But if we actually want to understand how the mechanisms work and what is happening behind the science of it, why is this outcome happening? Why is there a positive effect? We really go down to the research level where cellular studies happen, where they put curcumin in a cellular culture, see if the pathway that we are interested is in modulating or not. And if it is modulating, then is it creating a positive modulation or a negative modulation? For example, to test out inflammation, we do interleukin levels and we do inflammatory profiles, right? Now, we know that when we discuss senescent cells, we'll probably come down to that. And we know that senescent cells causes inflammation, right? So inflammation, if taking certain things like supplements related to cellular senescence, if that is reducing the inflammation, that means that link is pretty much proved. That, okay, I took this particular supplement to suppress my senescent cells, and the outcome should be what? That the inflammation reduces. Well, if the inflammation is reducing, then well and good, then that is what is working. And using these complex studies and research, basically experiment designs, there are existing research studies that can explain how these pathways are actually modulated and how one compound is directly related to a particular hallmark or not.
Wonderful. I really appreciate the formulations you are developing with Decode Age, all the products that are available that targeting nine hallmarks of aging. So coming to last three, we have 5 minutes. One is the stem cell exhaustion. What exactly it is? Are we exhausting all the stem cells by the time we age?
Right. It is actually I get a very usual analogy when I talk about stem cells, right? And we have already seen how a factory works. We have already seen how information is read. But with this particular hallmark, that is stem cell exhaustion, we need to think of the Reserve Bank of India, right? Where all the money is printed. Now, how the money is printed, right? If I have a 100 rupee note, then there is a chip for that 100 rupee note. And every time I want to start the production of 100 rupee note, I'll use that chip inside the machine and that will make the 100 rupee note. every organ in our body or every tissue in our body has this chip. It's basically the backup. If all the information is lost, if all the cells are lost, then how do I start reproducing those cells? So this chip is the master information. It will help us create more and more copies of the kind of cells that we need in our body. Now, stem cell exhaustion, when these chips are lost, there's a major problem. Like I'm kind of a modern person who watches TV shows. So if you see Money Heist, the TV show, right? What happens there is they don't try to steal all the notes in the country. They try to steal the chip in the company. So that's how important it is. And that's the stem cells for our body as well, or the our tissues as well. Let's say as simple as immune cells, right? When your immune cells are lost, let's say the immune cells are fighting a very heavy war, you got a COVID infection, and now all the immune cells are constantly being used up and destroyed while in that war. Now, we need more of these cells. So who do we go to? We go to the stem cells. We ask it to, start generating more and more copies so that more and more stem cells, so that more and more immune cells can be, made inside the tissue. Now, what if stem cell is not ready to do that? What if there is no stem cell to make that extra number of copies of the cell? Your entire immune system shuts down. If the same thing happens in the bone, your entire bone marrow mechanism, blood making mechanism shuts down. If the same thing happens in your brain, then the important cognitive reactions or the important neural networks that are formed in your brain, those shut down. If it happens in the heart, if the heart cells or the heart muscles does not have the stem cell to reproduce the muscle, then the tissue collapses, your heart collapses. It is basically the master chip in each and every organ and each and every tissue, which the day you are in emergency, this stem cell will come up. Okay, if you need more, I will make it. Similarly, if a country runs out of all 100 rupee notes, you go to RBI, you ask them to install that chip again, and you ask them to start producing the money. So that is what it is, basically.
Okay. So, and I think AKG, we already have the molecule, the product related to AKG, right? Like
yes,
stem cell exhaustion. It's a very well-studied molecule. Quite a good research is happening in this area as well. Let's talk about the cellular senescence. I think that's my favorite topic nowadays.
Right. So cellular senescence, we have to go back to the telomeres again, We back when we were discussing telomeres, we told that every time the telomere is cut off, the cell gets a signal to die, right? But that does not happen immediately. As soon as the telomere is gone, the cell does not die. It starts dying, right? It starts dying. Now, what does dying means? There is a certain phase of that time when the cell is dying, doing bad things to the body, and then it is completely dead. Now, that particular stage of cell, when it is dying, not dead, it's called cellular senescence. Even scientists today use a very unique term to, describe this stage of cell, which is called the zombie cell. Why zombie? Again, not dying. It's dying, not dead. So a zombie, and why zombies for the second reason is because when a zombie bites a healthy human, the healthy human becomes a zombie. Similarly, when a zombie cell comes in contact with the healthy cell, it becomes a zombie cell as well. So this creates a chain of events, creating a lot more zombie cells. Now, why are these zombie cells bad? They do something called SASP, senescence associated secretory phenotype. Now, senescence we know, associated we know. This, research papers have a habit of complicating stuff, but yeah, we'll try to simplify it. So senescence, we know that it's a zombie. Associated means it is associated to that. What is that association? That is the secretory phenotype. Secretory means it is releasing certain things that is going to change the phenotype, which is the structure or the functioning of the tissue. Now, that secretory word is very important because when the cell is in the zombie state, it releases a lot of inflammatory cytokines. Now, inflammatory cytokines is creating a lot of inflammation. It calls in your immune system. There is a lot of damage going around. Everything is in a haywire mode. This is basically the most worst scenario that happens with cellular senescence. When your stem cell exhausts and the immune cells cannot form, there is inflammation. When cellular senescence happens, there is a lot of inflammation. And there's already regular inflammation going on in your body while your body is trying to fight the infections. all this inflammation together comes up, and it comes into a single umbrella of something we call inflammaging. Because all these are aging associated mechanisms and they generate inflammation, it automatically becomes something called inflammaging. It's one of the most interesting topics to read about if you're really interested in the science, actually. So yes, zombie cells, inflammation, and diseases. That's how the series goes on.
And interestingly, good research is happening in this area. There is ongoing clinical trial, and people are waiting for the new movie release. But me and Darshit, we talk about when the results are going to get released for this particular clinical trial. We are waiting for the results. So coming back to the last hallmark of aging, which is altered intracellular communication. Now, again, just to give the revision about it, we talked about the genomic instability, which is the genome itself, what is surrounding that epigenetic. We talked about the shoelace and the telomeres associated with that. We talked about the origin and the nucleus and the mitochondria and how the evolution happened, and that's why mitochondrial dysfunction. We talked about the QC mechanism of loss of proteostasis. Then we talked about the nutrient sensing, that I'm hungry. Right now, I'm actually hungry. So at the cellular level, the nutrient level, how it's happening. We talked about the origin of cells, the chip and the RBI, stem cell exhaustion, and the money heist effect over there. And then we talked about the zombie cells, the cellular senescent cells, and they are getting accumulated, and somewhere we need to remove them. We do have Decode Age, we do have product for that as well. So coming to the last thing, altered intracellular communication, that why they are talking to each other now.
Right. So if we see all the hallmarks, right, the first few, like the genome, the telomeres, and the epigenetic, and the loss of proteases, that was all happening within the cell. Then we moved a bit outside. We saw that cellular senescence, stem cell exhaustion is kind of affecting the surrounding cells and tissues and organs. Now, altered intercellular communication, let's come back to the organism level. Let's come back to the conscious level. We all know there are hormones being exchanged day and night throughout the body that, okay, it signals some certain things to do certain things. Why is thyroid a age-related problem in women in India? Why? It's because the intercellular communication, the hormonal communication that was happening has now shut down. Not entirely shut down, but has been disrupted to a certain extent. Similarly, there are a lot of hormonal interconnections. Certain things secreted from the pituitary of your brain goes through your blood, goes to certain organs, tell the cells that, okay, dude, you have to do certain things like this to maintain the healthy cellular function. But that does not happen. Now, before Decode Age, when I was working in academia and all that, I did not understand this. But as soon as I founded Decode Age and I started maintaining a team, started to run an organization, communication was the most important thing throughout the journey. If the communication is not happening rightly, teams dysfunction. And when that kind of dysfunction happens, whatever roles or objectives the person had to achieve does not achieve. If the person sitting at the top, right, at the brain level, or the certain things that's communicating to everyone, if that person is not doing the right way of communication, then the entire flow is disrupted. Similarly, cellular communication is a very important factor in our body. Each cell communicates with the other. If they are adjacent to each other, they literally have networks between them to communicate with each other. If it's a neuron, that is a nerve cell, then there is literally two cells are like this, while there is a gap in the between, and the communication is happening through electric impulses. There are various ways your cells are communicating with each other. When this gets disrupted, when communication gets disrupted, the system disrupts. When the system disrupts, the organism disrupts. It's as good as running a company. It's as good as running a body. It's the two same things, actually.
Nice. What an analogy for an entrepreneur over here. Good. Now, before we close, your hallmark of aging number 10, 11, 12, after 4th of July, what's happening in there? What is it like? Why new hallmarks are coming up? Do we need so many hallmarks?
So hallmarks, it's an evolutionary study, to be honest. We cannot really say like which hallmark is directly related to aging or not. Because like we just said, telomere attrition also leads to genomic instability. Then why is genomic instability in itself a different hallmark? Because there are different mechanisms by which that is also getting affected. One thing leads to other. That's what a body is. That's what a system is. And we can never say that, okay, these 9 distinct reasons are why your body ages. There's always an intercommunication between all of them. And the 10th hallmark, people have been discussing different things, like inflammaging itself is trying to come out as a different hallmark, because chronic inflammation is one of the major problems in aging and age-related diseases. There is something called advanced glycation end products, which I think should be the 10th hallmark personally. Why? Because it's again a very different kind of mechanisms. Just like we saw all these 9, it's a very different and distinct mechanism. Advanced glycation end products is something I think is very interesting. Why? It's because your body has 2 3 things that it produces regularly. One says proteins and one is the sugars and all that, right? When sugar binds with sugar, your body can understand, okay, these are 2 sugar people coming, I need to break it down, right? When 2 proteins bind to each other and come, your body understands, okay, these are 2 proteins coming from the same family of biomolecules, I need to break it down. Body has a problem when a sugar and protein molecule comes together. We talk about diabetes a lot in India, at least, and one of the major molecular or molecular problem rather in diabetes is actually advanced glycation end products. Why advanced glycation end products? This process of a sugar coming together with protein and merging into a single complex, it's basically glycation. And these things cannot be removed again. This is like the protein thing we were talking about, that the bad protein accumulates. When such kind of complex systems come to your body, it cannot remove it, and that starts accumulating in your body also. Removing ages or removing the advanced glycation end products, and many research studies have shown extensive results on health and lifespan extension. So I believe that could be the next hallmark of aging. But obviously there are others being talked about, like the stiffening of extracellular matrix, So all the cells exist on a certain kind of matrix, just like the movie Matrix. All the elements are into a single matrix. So these matrix, when this matrix gets disrupted or it becomes stiffened, the cellular communication cannot happen. It's still very much related to altered intercellular communication. So I believe that advanced glycation should definitely rank higher above this one, is what I think.
Correct. And then the product that we have it with Decode Age is called Longevate. So is it taking care of multiple hallmarks of aging? How you came up with that molecule?
So we followed a lot of research papers actually, and a lot of cellular, animal, and clinical trial studies to understand how these works. And we have tried to come to a perfect blend where maximum hallmarks are addressed or taken as a target. I wouldn't say target because we are not actually making drugs out here, but still the idea behind this formulation is to have as many hallmarks into control. And it is important that we select components that can enter inside the cell, because things like curcumin wouldn't enter the cell. That is why it's in the epigenetic regulation and not within the cell, right? Or the extracellular communication where it is required. So certain things like now NAD plus is what's required inside the cell, but we don't see NAD plus anywhere around the circle. It's because NAD plus is too large to enter the cell. NMN in fact can enter into the cell, convert itself to NAD and go to both mitochondria and the genetic material. So that's how it goes about. We need to select the right components, the right compounds that can target the right things inside the cell and actually go inside the cell. You were also talking about blood-brain barrier and how we can cross that. So there are very few molecules that can actually do it, and resveratrol, terostilbene are 2 of them which can actually enter the blood-brain barrier, go into the brain and help with the neurotoxicity. So the idea was this only, to select the right components and the right synergies. Synergies is something we call like 1 + 1 = 3 kind of a thing. So right synergies, when 2 things come together, they give an extra effect. For example, like taking NAD plus or taking NMN to get NAD plus in your mitochondria might not be the only option, because like I said, it was a 5-step system. If we just try to target one step and forget the other steps, it's not going to complete the cycle, right? So to complete the cycle, we have to take it with CoQ10, which forms the 4th step into the entire respiratory chain. So all the things have to come together, to eventually give you the response. So that is why synergies are important. That is why blends are also important.
Perfect. See, we are running short of time. So what's our plan is to talk about every supplement or every product that is available with the clinical trial back data and what's the research that is happening, and come up with the webinars in future. One note I really want everyone to, those who are here and also people, those who are either on YouTube Live or those who will be listening to this webinar in future, is we definitely know, how good our car is. I do so much of research on my finance, my investment, my savings, and what all I should be doing. But we really, really forget to learn about our own body. We don't want to listen about mitochondria. We think it is very boring and very time consuming to understand DNA and nucleus. And I think it's very important. And we need to make that shift. We don't want to wait till we are deceased and we have problems and then we go to doctor and then we try to solve the problem. There is something that we can do it today. You don't have to take only the supplement. Those are things are good. I take it, but there are so many mechanisms that are being developed to work on all these hallmarks. It's quite a good research, and our efforts, like I think Decode Age, what I like about all of you is all these educational activities that we are doing to go to the basics, understand every step. Once we have this knowledge, and tomorrow you take any supplement, tomorrow you take that tea with antioxidants, or probably tomorrow you go for a specific cardiological, exercise, cardio specific exercise. If you do, you know what changes it can do to your body, and then you can positively do it. There was one question by Padma about the stem cell survival. Does stem cell survive only in hypoxia?
Well, that's a very specific question. I think that would have to be answered following a few research papers to be gone through. So yeah, no information to be shared without enough backup of knowledge.
Sorry about that. I just, because I read about it only recently when there was a paper about breath regulation and yoga and how it activates stem cells from a hypoxic level where they are. It gets them into the blood when they, when you hold breath. That's the idea behind it. So that's why I thought, is it so that stem cells only survive in hypoxic conditions? And that is a question.
I think that is something I have not come across, but I'd love to, look at the paper that you're talking about to understand which tissue are they actually talking about. Because, like I said, stem cell exists in different tissues. In fact, all the tissues of your body. So we need to consider if it was a tissue-specific study or, a generalized study to understand it better. Yeah.
Sure.
So please share the reference with us. We'll study that and we'll include when we are discussing more about stem cell exhaustion and molecules.
We can get in touch with you personally also to discuss.
Yes, that will be super interesting.
Yeah.
Sure. Thank you.
Sure. Thank you.
Good. And Darshit, thank you so much. I'm going to keep this recording forever, and I'm going to listen to it whenever if I have doubts regarding every basic mechanism. And is there anyone who wants to? So thank you so much. This was really, really, really helpful, and I needed this complete session. Okay, Varsha is there? Anyone wants to Parth? Any note from your end?
Any closing statements?
So this was a very great session on hallmarks of aging. And from this, I would like to ask Darshit to write a book on hallmarks of aging, as it got a very good explanation and people need this kind of easy explanation in their hands.
Right. I think I can do my best. I can at least try.
Yeah, Darshit, you have one more task to do now. Okay.
Yes.
Thank you so much, everyone.
No, I think there's a question from Ankur, and that is rather what everyone wants to ask. I think the mythology. So Ankur is asking, when can we expect a single miracle pill who covers all these aspects? So I think, Ankur, what has happened is mythology has trained us to be, believe that a single pill can help us. फिर वो सोमरस ही क्यों नहीं हो? सोमरस पी लेंगे तो सब हो जाएगा. But वो नहीं होगा ऐसे कभी. There will never be a single.
Somras is alcohol, no? I don't know.
Sorry?
My mythology is bad. So somras is alcohol. Amrit is the magic pill.
Oh yeah, so my mythology is also weak because I cannot stop criticizing it anyway. So the thing is, yeah, there will never be a single pill, Ankur. If there is ever a single pill, probably they are trying to scam you because the aging is not happening purely by these 9 hallmarks as well. There are a lot of other factors involved. This is just the cellular level. When we consider an organism, it's more complex than a universe itself, right? If we ever believe that a single pill can help us do all this, it will never. Not even single pill. You take 10 pills, it still won't happen if you're not maintaining the right lifestyle, if you're not doing what your body needs. Because your body is not used to pills, it is used to exercising. The way we have evolved in the last 70 years, if you actually see back in 1800s, late 1800s or early 1900s, people used to struggle. People used to do real physical work to get about to do something in their life. In the past 70 years, we have completely forgot that. Our lifestyle has become rudimentary. You want to sit down and get everything here itself. Well, that's not how we are designed. We are designed to function both physically and mentally, and until and unless we start doing that at an optimum level, no single pill is going to help us cure aging or reverse aging. There are a lot of things people do. I think Rakesh here himself, he's an athlete and he would be able to tell you what all kinds of things you can do in your lifestyle. The goal should not be to take that one single pill. Rather, the goal should be to avoid that single pill. And that is what we are on to as Decode Age as well. Because supplements itself is a word that means in the English dictionary to support something. And that something, if that is not incorporated, what are you supporting?
And I'll add one more thing, Darshit, over here, that if you look at overall the drug discovery evolution that is there, we talk about the tuberculosis, age-old disease. Even for that, we do not have a single pill, right? We have a combination because we want to target the organism from multiple pathways. If we talk about cancer, there is a protocol that is available. If we take the example of even HIV infection, there is a combination of drugs that are available. For even for diabetes, we give the fixed drug combination. So till date, I don't know if anybody knows that we are not able to come up with one magic pill that is able to solve the problem for the bacteria or for the virus. Yeah, we talk about the vaccines a lot. But we don't. And I agree for the lifestyle changes. Doctors keep talking about the lifestyle changes, but what are those lifestyle changes that we need to do? Available on internet, but Rakesh is the best guy to give you the protocol. You can just simply follow it and you can forget about the research in the background. So Rakesh, you should have a webinar like this to talk about all the biohacking protocols.
Yeah, so definitely a session on biohacking could help everyone here.
Right. And it actually brings me to like one single statement, like prevention before prescription. So we don't want the prescriptions of medications. We want the prevention to be done, and that's what we should focus on. But yes, Rakesh will be happy to take some lifestyle tips from you.
Surely looking forward.
Yes.
Any more questions from people, those who are here? Okay, so I already have to message from Decode Age. We are already it's 12:20. So let's carry on with our weekend. And thank you so much. It was really great discussion. Okay, let's connect again.
Thank you everyone. Good
Thanks
day.
everyone. Bye-bye. Bye.
Thank you.
Thank you.